Hello students, myself Gaurav Yadav pursuing PhD in microbial technology. So today's topic of discussion is plant tissue culture medium. What is the different components of plant tissue culture medium and what are the properties of those medium and the ingredients used in the plant tissue culture medium. So please watch my video till the end and do not forget to subscribe and like my channel. To start from the introduction part. What is plant tissue culture medium? The plant tissue culture and cell culture describe the sterile growth and multiplication of plant cells, tissues, and organs in vitro technique. Plant, plant cells cultured with nutrient medium in an artificial environment can be clonally propagated at a scale to more quickly produce mature and disease-free plants. High quality, uniform planting material can be readily propagated for plant application in molecular, genetic engineering, plant breeding, horticulture production, and environmental conservation. Such materials can also be used for plant sciences, phycology, and pharmaceutical research. Optimal growth and morphogenesis of tissues may vary for different plants according to their nutritional requirements. Moreover, tissues from different parts of the plant may also have different requirements for the satisfactory growth. There are some examples of plant tissue culture medium. First is white medium, was based on Uspenski and Uspenka's medium for algae. Gothret medium was based on knob salts. What are the knob salts? Knob salts is an aqueous solution containing the major inorganic constituents required for the growth of the cell of higher plants, namely calcium nitrate, potassium nitrate, magnesium sulfate, potassium dihydrogen phosphate, and potassium chloride. Basic medium that are frequently used are Mauritius and Skog medium, Linsmeer and Skog medium, Gamberg medium, and niche and niche medium. These are the basic medium which are frequently used in the plant tissue culture technique. Composition of the plant tissue culture medium. The plant tissue culture medium should generally contain some of all of the following components, which are as follows: macronutrients, micronutrients, vitamins, amino acid or nitrogen supplements, carbon sources, undefined organic supplements, growth regulators, and solidifying agents. According to the International Association for Plant Physiology, the elements in the concentration that are greater than the 0.5 millimolar are defined as macroelements, and those requiring the concentration less than 0.5 millimolar are known as microelements. It should be considered that the optimal concentration of each nutrient for achieving maximum growth rate varies among the plant species. Essential elements in plant cells or the tissue culture medium include that are considered as macronutrients are carbon, hydrogen, and oxygen. Other macroelements include nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur for the satisfactory growth and morphogenesis of the plant. The optimum concentration of phosphorus, magnesium, sulfur, and calcium ranges from the 1 to 3 millimolar. The culture medium should contain at least 25 to 60 millimolar of inorganic nitrogen for satisfactory plant growth. Potassium is required for the cell growth of most plant species. Most medium contain potassium in the form of nitrate chloride salts at the concentration ranging between 20 to 30 millimolar. Is micronutrients. Now, essential micronutrients are the important for plant growth and tissue growth. These include iron, manganese, zinc, boron, copper, and molybdenum. Iron is usually the most critical of all the micronutrients because iron helps in photosynthesis and plant growth as well. Copper and cobalt are added to the culture medium at the concentration of 0.1 micromolar. Iron and molybdenum at the concentration of 1 micromolar. Iodine at 5 micromolar. Zinc at 5 to 30 micromolar. Manganese at 20 to 90 micromolar, and boron at 25 to 100 micromolar. The element used as either citrate or tartrate salts in culture medium. However, there exists some problem with this compound for their difficulty to dissolve and precipitation after medium preparation. There has been trial to solve this problem by using EDTA. A procedure for preparing an iron chelator solution that does not precipitate have been also developed. Cobalt and iodine may be added to the certain medium, but their requirement for cell growth has not been precisely established. Sodium and chlorine are also used in some media, in spite of report that they are not essential for the growth of the plant species. I'll discuss about the carbon and different energy sources. These include lactose, galactose, maltose, starch, sucrose, glucose, and fructose. In plant cell culture medium, beside the sucrose, frequently used as a carbon source at a concentration of 2 to 5%, other carbohydrates are also used as and they were reported to be less effective than either sucrose or glucose. The latter was similarly most effective than fructose, considering that glucose is utilized by these cells in the beginning, followed by fructose. Sucrose was reported to act as a morphogenetic trigger in the formation of auxiliary buds and branching of adventitious roots. It was found that supplements of sugar, cane molasses, banana extract, coconut water to the basal medium can also be good alternative for reducing medium costs. These substances, in addition to sugar, they are the sources of vitamins and inorganic iron which are required for the plant growth. The human body requires vitamins for their growth, so plant also require vitamins for their proper growth. Thiamine, it also helps in the rapid cell growth which require only 0.1 to 10 mg per liter of thiamine. Nicotinic acid, that is required in 0.1 to 5 mg per liter of growth concentration. Pyridoxine, which is required in 0.1 to 10 mg per liter concentration. Myoinositol is added in small quantities to stimulate cell growth of the most plant species. It is generally used in plant cell and tissue culture media at the concentration of 50 to 5000 mg per liter. Some plants are able to synthesize the essential requirements of vitamin for their growth. Some vitamins are required for normal growth and development of plants. They are required by plant as a catalyst in various metabolic processes. They may act as a limiting factor for cell growth and differentiation when plant cell and tissues are grown in vitro. Nicotinic acid and pyridoxine, however, not a not an essential for cell growth of many species. They are often added to culture medium. Other vitamins such as biotin, folic acid, ascorbic acid, pantothenic acid, tocopherol, riboflavin, p-aminobenzoic acid are used in some culture medium. However, they are not growth limiting factor. It was recommended that vitamins should be added to culture media only when the concentration of thiamine is below the desired level or when the cells are required to be grown at the low population densities. Myoinositol is believed to play a pivotal role in cell division because of its breakdown to ascorbic acid and pectin and incorporation into phosphorinositides and phosphatidylinositol amino acids. As we all know, amino acids are the building block of protein. The plant require amino acids for their optimal growth and are usually synthesized by most plants. However, the addition of certain amino acids or amino acid mixtures is particularly important for establishing cultures of cells and protoplasts. Amino acids provide plant cell with a source of nitrogen that is easily assimilated by tissues and cells faster than inorganic nitrogen sources. Organic supplements. The one of the undefined organic supplement is activated charcoal. The addition of activated charcoal is sometimes added to the culture medium where it may have either a beneficial or deleterious effect. Growth and differentiation was stimulated in orchids, onions, and carrots, tomatoes by using activated charcoal. On the other hand, activated charcoal inhibited the cell growth of soybean plant. The presence of 1% activated charcoal in medium was demonstrated to largely increase hydrolysis of sucrose during autoclaving, which causes acidification of culture medium. Some medium are supplemented with natural substances or extracts such as protein hydrolysates, coconut milk, yeast extract, malt extract, ground banana, orange juices, and tomato juice to test their effect on growth enhancement. A wide variety of organic extracts are now commonly added to the culture media. Explanation of the mode of action of activated charcoal was based on absorption of inhibitory compounds from the medium, absorption of growth regulators from the culture medium, or darkening of the medium. Agents. As we all know, the solidifying agent play a crucial role in microbiological culture medium. They also play a pivotal role in plant tissue culture medium as well. The agar is the only acceptable polysaccharide, which was obtained from the seaweeds, is the universal solidifying agent which is used for the plant tissue culture medium in the at the concentration of 0.8 to 1% only. Hardness of the culture medium greatly influences the growth of culture tissues. There are a number of gelling agents such as agaros and gellan gum other than the agar. Agar has several advantages over other gelling agents. That's why we use agar. First advantage is it can be mixed with water. It easily melts at the temperature ranges 60 to 100 degrees Celsius and solidifies at approximately 45 degrees Celsius temperature. And it forms a gel stable at all feasible incubation temperatures. Agar gel do not react with media constituents and are not digested by the plant enzymes. It is commonly used in medium at the concentration ranging from the 0.8 to 1.1%. Sorry, 1%. Now come to the growth factors. They are generally classified into the following category: auxins, cytokinins, gibberellins, and abscisic acid. Moreover, proportion of auxin to the cytokinins determine the type and extent of organogenesis in the plant cell culture. There are some auxins which are natural such as IAA, IBA, 2,4D and NAA. But there are also some synthetic auxin which are also used in the plant tissue culture medium. Plant growth regulators are important in plant tissue culture since they play vital roles in stem elongation, tropism, and apical dominance. Indole acetic acid is only natural auxin occurring in plant tissues. In tissues culture, auxins are usually used to stimulate callus production and cell growth, to initiate shoots and rooting, to induce somatic embryogenesis, to stimulate growth from shoot apices and shoot stem culture. Second one is cytokinins. Cytokinins prove to stimulate cell divisions, induce shoot formation and axillary shoot proliferation, and to retard root formation. Cytokinins are relatively stable compound in culture media and can be stored desiccated at 20 degrees Celsius. DMSO has an additional advantage because it acts as a sterilizing agent. Thus, solution containing DMSO can also be directly used in the culture medium containing cytokinins. So that's why we dissolve our cytokinins in the DMSO. But cytokinins are frequently reported to be difficult to dissolve, and sometimes addition of few drops of 1N-HCl or 1N-NaOH facilitate their dissolution. Otherwise, as I told you, we can use DMSO for the solubility of cytokinins, which also protect plant from the other injuries. Gibberellins. Gibberellins comprises more than 20 compounds in which GA3 is the most frequently used gibberellin. These compounds are proven to enhance the growth of callus and also help the dwarf plant's elongation. Other growth regulators such as abscisic acid are also added in plant tissue culture medium that is usually supplemented to inhibit or stimulate callus growth depending upon the species of plant. It enhances it enhances shoot proliferation and inhibits later stages of embryo development. Although growth regulators are the most expensive medium ingredients, they have little effect on medium cost because they are required in very small concentration. I would like to discuss how we can prepare plant tissue culture medium. It is slightly different in comparison to the microbiological culture medium. A convenient method for preparation of culture medium is to make concentrated stock solution which can be immediately diluted to perfect concentration before use. Concentration of inorganic and organic components of media are generally expressed in mass values. In the International Association for Plant Physiology has recommended the use of moles value. The solution of macronutrients are better to prepare in stock solution of 10 times strength of the final volume. The stock solution can easily be stored at the refrigerator at the temperature 2 to 4 degrees Celsius. Micronutrient stock solutions are made up at 100 times of final concentration of working medium. The micronutrient stock solution can also be stored at the refrigerator or refrigerated until the needed. Iron stock solution should be 100 times concentrated than the final working medium and stored in refrigerator. Vitamins are prepared as either 100 or 10,000 times concentrated stock solution and stored in freezer until the use. It is desired to keep them for long. They can be refrigerated for 2 to 3 months and should be discarded thereafter. Stock solution of growth regulators are usually prepared 100 to 1000 times the final desired concentration. This is the medium composition. As you can see there, this is the list of different medium as I told you in the introduction part. These are the macronutrient list, these are the micronutrient list, these are the vitamins and other supplements, these are the concentration which can be used according to the different medium. The main important question arises here is that how we can select our media. So for this, for the establishment of new protocol for specific purpose in tissue culture, a suitable medium is better formulated by testing the individual addition of a series of concentration of a given compound to a universal basal medium such as MS, LS, and B5. The most effective variables in plant tissue culture medium are the growth regulators, especially auxin and cytokinins. Full strength of salts in medium proved good for several species, but in some species the reduction of salt level to half the full concentration gave the better result in vitro growth. Sucrose is often assumed to be the best source of carbon for in vitro culture. The level used from 2 to 6 gram per liter, and the level must be defined for the each species of plant. The student have any query related to plant tissue culture medium or other things, you can ask in the comment section, and please do not forget to subscribe my channel, and please do like my video and share my video. Thank you.